Hey, welcome to The Explainer. Today, we're diving into something pretty mind-bending, a field that's moving beyond just reading the blueprint of life to actively writing and editing it. Yeah, we're talking about synthetic biology.
All right, let's just jump right in with a huge question. Forget about coding on your computer for a second. What if the programming language was DNA and the computer was a living cell?
Think about it. We could build these tiny, microscopic factories that could do incredible things, like fight diseases from the inside out, create sustainable fuels from waste, or even grow brand new materials that are stronger than anything we can make ourselves.
And look, this isn't some far-off sci-fi fantasy. No way. This is the revolutionary field of synthetic biology, and it is happening right now.
Okay, so let's get down to it. What exactly is synthetic biology? Let's break it down.
So at its core, synthetic biology, or SynBio for short, is all about applying engineering principles to biology. The big idea here is to stop just studying life and to start building with it. It's like taking the same design and build mindset we use for bridges or software and applying it directly to living cells.
All right, so if you're going to engineer life, you're going to need a pretty special toolkit, right? Let's open it up and see how this all actually works.
Now, I know what some of you are probably thinking. Wait, isn't this just GMOs with a new name? And that's a fair question, but there's a really key difference. Traditional genetic engineering is usually about moving a single existing gene from one place to another. A great example is putting the human insulin gene into bacteria so they can make it for us. Synthetic biology, though, it's a whole different level. It's more like building an entirely new app for the cell from scratch, using multiple genes of standardized parts. We're talking about creating whole new pathways or circuits that can make a cell do something totally new, like blink on and off.
And this didn't just happen overnight. The groundwork was laid way back in 1953 when we first figured out the structure of DNA. But just look at how fast things have picked up since then. In 2000, we built the first simple biological circuits. A mere decade later, we created the first cell running on a completely synthetic genome. And then, bam! 2012, CRISPR comes along and just changes the entire game with its power and precision.
So what's powering this whole revolution? Well, it really boils down to a few key technologies. First, our ability to write DNA through synthesis and read it through sequencing has gotten insanely cheap and fast. Then you have this brilliant idea of modularity, creating standardized parts, kind of like Legos, that you can just snap together to build new biological systems. And of course, the big one, CRISPR. It's basically the ultimate find and replace tool for the genome.
So, with this incredible toolkit in hand, scientists are doing some absolutely mind-blowing stuff. Let's look at how biology is being totally reimagined.
And this isn't some niche thing happening in a few university labs. Not at all. We're talking about big business. To put it in perspective, the global market for this was already worth almost $4 billion way back in 2016. That kind of money just fuels even more innovation.
And the applications? They are everywhere. It's stunning. We're talking about engineering microbes to hunt down cancer cells, producing life-saving drugs in yeast, creating self-healing concrete, growing meat in a lab, even designing bacteria that can sniff out landmines by glowing. See, synthetic biology isn't just one thing. It's a platform technology that could literally change everything.
Okay, so how does this actually work in the real world? Let's take a great case study, an anti-malarial drug called artemisinin. The old way was to extract it from a specific plant, which was slow and really expensive. But with SynBio, scientists basically designed the genetic recipe from the ground up, synthesized that DNA, and popped it into yeast. And now they can brew this life-saving medicine in huge vats, kind of like making beer. The result is a cheaper, more reliable supply of a critical drug. But with this much power comes a whole lot of responsibility, right? A technology like this naturally brings up some really big questions. So let's shift gears a bit and talk about the crucial ethical side of all this.
So the ethical debate here is really multifaceted. On one hand, you've got these huge philosophical questions. Are we playing God? Is it right to create new forms of life? Then you have the more practical concerns. What about biosafety? What if one of these custom-built organisms gets out of the lab? And what about biosecurity? Could someone use this to make a weapon? Now, on the other side of the debate, you have the counterpoints. People point out the incredible potential to solve massive problems like disease and hunger. They're also developing safeguards, like building in kill switches so these organisms can't survive in the wild. And, a lot of this research is already covered by existing regulations.
So, where are we headed?
What does our future look like in a world where we can truly engineer biology from the ground up?
When you boil it all down, we are learning to write the code of life. It's like we've been handed the ultimate programming language. So the real question for all of us is, what story will we choose to tell? Because this isn't just a story for scientists to write. It's a story for all of us.